So in the mitochondria, the couple electron-proton transfer through NADH guanine oxireductase enzyme use. So in this way, let's begin to understand this is the inner mitochondrial membrane and inside is the inner mitochondrial matrix. So remember, and in this way here is basically I am drawing the NADH dehydrogenase enzyme. Basically it is basically the protein containing enzyme containing protein, globular protein are present in the inner mitochondrial matrix and contain a Q pool. And this Q pool is the guanine pool. And here is the NADH dehydrogenase enzyme used to NADH dehydrogenation will lead to NAD plus, and in this way this is the oxidation stage, the oxidation. So the oxidation of NADH, and in this way here is you can understand this is the proton will move out too, and in this way the FMN is basically absorb the electron and the iron sulfur protein will transmit into the to the guanine to form a reduction into the QH2, and in this way the QH2 formation, and in this way remember the 5 proton is involving. In this way here is the FMN bridges NADH and 2 electron donor with iron sulfur protein 1 and electron acceptor also, basically with iron sulfur 1 protein containing electron acceptor and the L-shaped complex 1. Overall reaction we will discuss now. Here is the NADH plus guanine plus 5 proton. Here you can pump out and to form a NAD plus as well as the QH2, and 4 proton will move pump out. And in this way you can understand the flavin mononucleotide oxidized will reduce the 2 electron and 2 proton in the FMN. Here you can see this, and this will basically use to reduced FMNH2. So remember the FMNH2 is reduced, and in this way it will basically the transmit the electron to the guanine to form a basically it is ubiquinone which that's conferred. And here is the complex 3 and complex 2 is involved. First is the complex 3 is the cytochrome BC1 which that is involved, and cytochrome B will accept the electron while the pump out the proton. 4 proton, 2 will from the inner mitochondrial matrix and the 2 from the QH2, and in this way the cytochrome B transmit the iron sulfur again protein and cytochrome C1 is the mobile which that is not we are understanding, but here is the QH2 also receive from the iron sulfur protein containing succinate dehydrogenase enzyme containing channel here protein. The succinate convert into the fumarate, and in this the FAD and iron sulfur which that is from the FADH2 to give the 2 proton, and this 2 proton also from the cytochrome B which that will pump out, while again this is mobile protein. So remember the iron sulfur protein, and this is a succinate dehydrogenase enzyme, and it is contain iron sulfur protein and FAD. FAD basically conversion from the FADH2, and FADH2 will transmit the 2 proton to the QH2 to the QH2, and in this way the cytochrome will receive the electron will transmit to the 4th complex. This is the cytochrome oxidase. This cytochrome oxidase transmits the QA, cytochrome A and cytochrome A3 to QB, copper containing protein B, and in this way here is the half molecule of oxygen, and the 2 proton will lead to electron will also transmit to the reduction reaction to produce the water. This is the condensation reaction, while here is the cytochrome oxidase is involved. This is the electron transport chain, remember. So in this way the inner mitochondrial membrane is here. You remember that. So basically couple electron proton transfer through NADH oxireductase we had discussed, and we had discussed about the electron transport chain little bit.